Welcome to our channel. In this video, we are diving into the fascinating world of protein purification, specifically focusing on the first step, cell breakage and fractionation. These processes are crucial for isolating proteins from cells and tissues for further studies or research. Cell breakage and fractionation are fundamental steps in protein purification and cell biology research, and they are done for several key reasons. The first one is the isolation of specific proteins or organelles. Researchers often need to isolate specific proteins or cellular components like mitochondria, nuclei, or ribosomes for detailed study. By breaking open the cells and then using fractionation techniques, these components can be separated from the rest of the cellular material. Why? This isolation allows for study of structure, function, and interactions of specific proteins or organelles without interference from other cellular components. For the analysis of cellular processes, cell fractionation allows the scientists to analyze specific cellular processes in different parts of the cell. For example, researchers might study how enzymes work within mitochondria or how ribosomes are synthesized by the proteins. To perform the accurate biochemical assays, scientists need to pure samples of the molecules or organelles they are studying, that is, to find out their protein of interest. Fractionation techniques help achieve this purity by separating different cellular components based on their size, density, or other physical properties. By breaking open the cells and separating their components, researchers can study the structure of cell organelles and other subcellular components in detail. Many diseases are caused by dysfunction in specific cellular components. By isolating and studying these components, researchers can better understand the underlying mechanism of diseases. This understanding then can lead to the development of targeted therapies and diagnostic tools. The first step in protein purification is to disrupt tissues and cells in a controlled manner. This is typically achieved through a process known as homogenization. Using gentle mechanical processes or procedures, we can rupture the plasma membrane of cells, releasing their contents. There are several commonly used methods for homogenization. The first one is high frequency sound or ultrasound. Using this method, ultrasonic waves are used to break the cells open. Then we can also use mild detergents that make small holes in the plasma membrane, allowing the contents of the cell to leak out. You can also use high pressure methods like forcing the cells through a small hole using high pressure that causes the cells to break open. The last one is by shearing. Cells can be sheared between a close fitting rotating plunger and the thick walls of a glass vessels. There are also other ways for carrying out the process of homogenization. Once we have the homogenate, the next step is to separate its components. Centrifugation is the most widely used technique for this purpose. The homogenate is placed in test tubes and spun at high speeds in a centrifuge or ultracentrifuge. Modern ultracentrifuges can rotate at speeds up to 100,000 revolutions per minute, producing forces as high as 600,000 times gravity. One approach to fractioning the cell homogenate is differential centrifugation. This involves repeated centrifugation at progressively higher speeds. Each round of centrifugation separates components based on its size and density. Low-speed centrifugation sediments larger components like whole cells, nuclei, and cytoskeletons. Medium-speed centrifugation isolates mitochondria, lysosomes, and peroxisomes. High-speed centrifugation separates smaller vesicles and fragments of the endoplasmic reticulum. And lastly, very high-speed centrifugation isolates smallest components like ribosomes and large macromolecules. In velocity sedimentation, the subcellular components are separated based on their size. The homogenate is layered over a dilute sucrose gradient and then centrifuged. As components sediment at different rates, they form distinct bands, which can be collected individually. Equilibrium sedimentation, also known as density gradient centrifugation, separates components based on their buoyant density. The sample is centrifuged through a steep gradient of sucrose or cesium chloride. Components migrate to the region where their density matches that of the gradient, forming distinct bands that can be collected later. By using analytical techniques like Western blotting or mass spectrometry, one can confirm the presence and purity of target components. Understanding these basic techniques is essential for any researcher working with cell extracts and protein purification. By mastering the cell breakage and centrifugation, you can isolate and study specific proteins with greater precision.